Hi, good morning, and welcome to the ZP Developer Zone webinar. So we do this every Thursday at approximately 8 a.m. London time, though I must admit we do go early when there's a lot of material or meetings have come up, and so I have gone early because there is actually today a lot of material and a lot of meetings. But let me go forward very quickly. Say, don't forget the ZP Academy. It's a free website where you can find at least 2 free courses on ZP, or really on electrochemical biosensing. So ZP Academy has at least 2 free courses on it. Don't forget our webinars. We do have a webinar, for example, in an hour and a half from now on food sense, which is describing one of our sort of technology branches or one of our product branches. We do have collaborations. In fact, I've just had emails this morning from Ireland and from the UK asking for collaborations, which we do. In fact, we just had some more people asking for jobs in Norway, and I saw my team responding to those people there. And we do have our ZB Developer Zone, and we do have our workshops. I think the workshops are probably one of the best ways of getting information. Many of these questions are actually related to workshops. So we do in-person workshops. If you see a link jump up, I've already put the links underneath the YouTube video. So if you're going to our website and you're watching the video, I also know that these links are underneath the YouTube video as well. So this is recorded and put underneath, and any links are already on the sort of in the description box on the YouTube channel. We do have a workshop in November in Norway. This is probably one of the best ones because it's a 2-day workshop. We do have a 1-day workshop in Coventry in November, and we do have a workshop also in Coventry in December. The reason we have such a frequency of workshops is because then nobody's kind of waiting too long to essentially come to one of our workshops. You know, there's at least one a month, and you can see here there's 2 in 1 month here, and then one in December as well. We are having our Scandinavia Sensor Summit. We're going to do a one in the UK and one in Norway. S Cubed, the one in is open for early bird registration, and it will be on the commercialization of electrochemical sensors. The one in Norway in January 2025 will be on precision agriculture. Just remembering myself. I did say that we do a lot of free webinars. There will be a link in a minute. The link is already underneath the video on YouTube, but it's probably worth saying that these are happening in November and December. We do have one on Julie. Julie, most of our technologies these days actually can plug into Julie. It's a cloud data management system. It won't lock your data up. You can download your data, or you can get your data via what's called an API, an application program interface. That said, there's a webinar on it anyway. There is a Sense It All webinar as well, and there's also a Food Sense webinar very shortly as well. I want to say thank you to Plub Echo for coming on this morning, and I will go forward a little bit now. So here's the link. You can find those free webinars. Those webinars are definitely worth attending. Corporate workshops. This is something last week we were traveling, and actually we were doing an on-site workshop for one of our large corporations. You can understand that it's quite an expensive effort to go on site with somebody and take all our equipment, and we take an extra engineer as well. So that's quite an effort, let's say. So they really, I call them corporate because they're quite expensive, but we do have corporate workshops. Questions for this week, materials for CGM developers. That'll become explanatory as I go through it. There's a lot of glucose involved this week. Somebody else, I want to explicitly say now, this person wants to calibrate their NO sensor, their nitric oxide sensor, their hydroperoxide sensor, their ATP, adenosine triphosphate, and their pH sensor. This person who's asking this question, they've actually bought quite a lot of stuff from us. When you have, and stuff is a terrible term, but when you have bought this kind of material, I'm explicitly saying you can come to one of our workshops for free, because I feel like you've bought a lot of material, you have a lot of questions, and it'll be easiest to get you on site in our labs and actually give you a bit of, you know, one-on-one training. So I do hope that you will come to one of our workshops. And as I say, explicitly for you, number 2, there's a free workshop. You can come to one of our workshops for free. Number 3 is about building immunosensors. They want to know really how to block sensors to stop non-specific binding. And they also want to know a little bit about impedance spectroscopy. I mean, that is covered in our workshops, but I'll share some data today. Question number 4 is somebody in India is interested in our pH and oxygen sensors and also our electronics. It's probably worth saying with our electronics, the world of electronics in electrochemical biosensors has really changed. So there's no like preamplification and then we read the signal. It all happens on the electronics. Now, obviously within the electronics, there's all sorts of amplifiers going on, but that's essentially a black box. You know, it's the electronics. It's almost like single chip analog front end these days. I think the world of the days of making discrete electronics or discrete PCBs for measuring electrochemical biosensors, I think is over. There's so many good sort of chip scaled chip scale potentiostats these days that it's kind of not necessary. Somebody else wants to measure potassium sensors in blood, and I will comment upon that. The one thing I would say is that I will mention it. We have technology readiness levels, manufacturing readiness levels, and what's called application readiness levels. I want to say very clearly that ZP will have a high technology readiness level and a high manufacturing readiness level, but your application is your application. And that's the bit that you really have to work upon. So, quick comment, our potassium sensors were designed for blood. Will I support you doing human studies if you just buy them off the website? I can't. I literally can't, because the sensors on our website are intended for R&D purposes only. They're only really so you can do sort of proof of principles and lab tests. Once you start talking about blood, especially human blood, then you really have to have a very formal contract and budget with us. I can't support the testing of human blood on those sensors. Anyway, that said, question number 6, this person's also been invited to come free to our workshop because they're a significant client. The lactate formulations do have to be stored in the freezer. You can't take them in and out of the freezer. What I would do is if you've got a vial of lactate formulation, I know you haven't got much material, but you almost have to decant it into several vials so that you can take it out, use it. But if you freeze and take it out, use it, but don't put it back. What I'm saying here is the freeze thaw action of taking something out of the freezer, putting it back in the freezer, taking it out, putting it out. Obviously, ice crystals grow, ice crystals shrink, ice crystals or melt rather. That can end up denaturing proteins, and enzymes are just forms of protein. So we don't like the idea. Lactate in particular, the enzyme is particularly weak. So we don't like the idea of you freezing and unfreezing that several times. The sodium and potassium activation solutions, we just say keep them in the fridge. I mean, it's just keep them in the fridge just means they're kept in a cool, dark place. They're a lot less sensitive than the lactate formulation. Question number 7. Somebody had a whole list of sensors. They wanted 1 lactate sensor, 1 potassium, 1 cortisol, 1 testosterone, 1 histamine. I genuinely said to them, or I'm saying to them as well in there, just choose one of them to start with. Honestly, you're giving yourself 1, 2, 3, 4, 5 headaches. And I think you're kind of, the size of your team may be just one person, and it's too much to try and do all of this. You won't do any of it well. You have to do one thing. And this is free advice. The one thing you should do is potassium, because I know it's the most, of this list, it'll be the easiest for you to implement into a kind of working prototype or working minimally viable product. Question number 8, somebody has a pH sensor. That's right, they've got a pH sensor. They're testing it. They're getting something like 4 millivolts per pH, which is well below, it's an order of magnitude low. And I'll comment upon that. Question number 9 is a question, can they co-detect glucose and dopamine? Obviously, co-detect is a really, you can co-detect by having a glucose sensor and a dopamine sensor essentially next to each other in the same solution. You could also try to do it on one working electrode, which I suspect is what they're trying to do. So I will comment upon that as well. Now, so far, I've been speaking for approximately 10 minutes, and that's only going through the questions. So you can see why I had to go early this morning. Question number 1, I suppose it's worth saying that, at ZP, we are fairly, there's 2 types of technology that we often work on. Point of need or in vitro diagnostics. So, for example, in an hour and 20 minutes from now, we will be doing a food sense webinar. And that's a kind of point of need example where we take a sample of chili and ginger. We test the hotness of the chili and the pungency of the ginger. That's a kind of point of need. That's kind of like medical diagnostics. When you have a drop of blood, you do a test on it, you get a result done. That's a lot of what we do. The other part of what we do a lot of is also, I put here point of care. I'm really trying to say continuous monitoring. So at ZP, we were the engineering team behind a company called PK Vitality. Very, very good company. In the end, they weren't able to get their second round of funding, but ZP licensed all the technology, and this was a CGM technology. So our business model is contract development, contract manufacturing, and it's not always discrete testing. Sometimes it's continuous work as well. But a technology like this, where they're trying to do microneedle monitoring of glucose in interstitial fluid, which we were very involved with, it's a tough program. But these are the kind of things that we do at ZP. Now, the reason I bring that up is because I'm still talking about continuous glucose monitoring. This is a very different form factor for continuous monitoring. This one's actually set up for glucose and cortisol. This is a 100% ZP technology. We've got the sensors in here. We've got the board. Later on, I'll talk about the single-purpose biosensor circuit, which is relevant to people including pH and glucose. This is that, so I'd always call it generation 1, a single-purpose biosensor circuit. So it's in that little capsule there. We've got a couple of batteries, and we've also got a transducer for getting the information off the board and essentially into the cloud. These are slides that I use in our workshop. So if you're really unfamiliar or you want to really get a really good understanding of electrochemical biosensing, then this is one of the slides I use there. But the reason I bring it up now is because when you construct a CGM, especially these transdermal CGMs, transdermal just means obviously through the skin. You know, they're often constructed from a material such as a platinum wire. We'll put some insulator on there, and we'll put some silver chloride around that. So it's essentially 2-electrode mode. We then functionalize the platinum with an enzyme to make it specific to the enzyme of interest. So there's plenty of people out there now trying to build these CGMs. That's why on our website we hold material like silver chloride wire. Some people use silver chloride wire as their reference electrode when building these kind of transdermal wire CGMs. And I also have this kind of platinum electroplating solution because platinum is super expensive. So having platinum wire, we do use platinum wire, by the way, at ZP. It's really expensive. So people are very interested in then kind of reducing the cost and doing things like electroplating of platinum onto other materials. And we do have a solution for that as well. As these links appear, I've already taken these links and put them underneath the video, as I say, on the YouTube channel as well. This person is invited to one of our workshops. We do have one in November, at the end of November in Norway. I strongly suspect you should come to this. They're trying to calibrate their nitric oxide sensor, their hydroperoxide sensor. I think it's their ATP. They put OTP in the email, but I think they meant ATP, adenosine triphosphate, and also their pH sensor. The nice thing about ZP is I was able to kind of find their order quite readily as well. So I was able to kind of look at what they said in their email and then do a reverse kind of lookup using dates and stuff and actually find what they had ordered. I realized that maybe they didn't mean ATP, they meant ORP, so I haven't covered that today. We test these kind of sensors or calibrate these sensors either by doing drop testing or beaker testing. I think in this case, both work, by the way. Whether you do beaker testing or drop testing depends on the final application. If the final application is discrete testing, then drop testing is often more appropriate, and if it's continuous monitoring, then this kind of beaker testing is more appropriate. As I say, with this particular person, I would actually suggest you do come on a workshop. I'm not charging you for it, but you do have to get yourself to Norway. So there's flights and hotels. The food during the workshop is obviously free, and then the workshop itself would be free to you as well. The reason I'm sort of showing this is that if it was an NO sensor, nitric oxide sensor, the way we're kind of calibrating it is that we're adding a solution of nitric oxide, and I'll discuss that in a minute, to the sensor as it's running. We are running these nitric oxide sensors at it sounds a bit ridiculous, 1.05 volts. I say it's ridiculous because where did this 0.05 come from? But sometimes you just get caught in your own tradition. So we're kind of caught in our own tradition that we're adding nitric oxide to the sensor, and as we increase the concentration, the signal goes up. nitric oxide, here we are, 1.05 volts. The save rate of half a second is it's a bit silly. I think it should just be 1 second. Otherwise, it's completely fine. And literally, this is how we're testing it. I will talk in a minute about nitric oxide is quite hard. So in this kind of experiment, it's the same way that we would test and calibrate a glucose sensor as well, by the way. We have a break-in period. It's not unusual. Well, it is expected. If you come on one of our workshops, I'll explain why. It always happens. When the signal settles, that's when we start adding in the nitric oxide, and the signal increases accordingly. So as we add it in, and then we can work out our kind of, we'll do sort of a linear best fit. You don't have to do linear regression, but anyway, that said, we did do linear regression on this. And we use the potentiostat, but I'm pretty sure that they're using the same potentiostat as we are. I say put your nitric oxide is hard. It's hard for this reason. Luckily, this video is recorded because I had to literally take this from one of our lab scripts. So nitric oxide generation in the lab. So what you're going to try and do is you're going to try and make a saturated deionized solution of nitric oxide in the lab. And we're assuming from the saturation solubility of nitric oxide that we're making up a 1.8 millimolar. So we make up this kind of bulk of nitric oxide. You can see why we can't ship this solution, because nitric oxide is not super stable. It is not something that we can make up and ship. There may be better ways of making nitric oxide, but this is the way that we're making it. So what we're doing is we're taking a round bottom flask and we're putting in 17 grams of iron sulfate and 17 grams of sodium bromide and 8 grams of sodium nitrite. And all that is being done in a round bottom flask with 100 milliliters of DI water. Now, you have to do your own risk assessment on this. We've obviously done our risk assessment. You have to do your own risk assessment. I'm telling you a procedure. I'm telling you, you have to get a qualified person to check this procedure, and you have to do your own sort of research on this. So once these sort of iron sulfate, sodium bromide, and sodium nitrate are mixed in that 100 mils, it's starting to fume nitric oxide, which we then bring using a line across to a 1 liter solution of nitric oxide and collect the gas. And we're sort of assuming that we're saturating that solution with that gas. We've got chemistry background, so we do this. There's no sort of heating and stuff. It's quite straightforward to add these chemicals, but I don't think it's, for an engineer, this is quite a hard, let's say, thing to do, but this is literally how we do it. And then we have this 1 liter of solution saturated with nitric oxide, and it's that we're then dropping into this beaker in order to get these little staircases that I was describing earlier on. So we're taking that 1 liter saturated nitric oxide solution, only adding, as you can see, microliters to milliliters in order to then get these kind of stepwise concentration. So I do use the word hard here because it is hard. I mean, the easier way to do, by the way, is just get a nitric oxide cylinder. But nitric oxide is toxic. Getting a pure cylinder of nitric oxide, I think, is quite dangerous. People buy cylinders with nitric oxide blended in with a carrier, like I think probably like nitrogen. You can also try that as well. But it's not trivial working with nitric oxide. It's probably worth saying as well that I'm going to slightly change on to hydrogen peroxide as well. Before we ship the hydrogen peroxide sensors, we do a test here, and I forget the potential that we're applying here. It's maybe something around about 50 millivolts. And what we're doing is what we do. I'll in fact, I've got it written down here. It's minus 100 millivolts. So we're doing a chronoamperometric experiment. This is where the potential goes on, and we're adding. And what we're testing at 0.5 micromolar, 4 micromolar, and 10 micromolar. And I say it here that we've taken a 30% hydrogen peroxide solution and we've made up these solutions. The reason we're not shipping this is I think 30% hydrogen peroxide, which you can get from Sigma, which is quite stable. But if we made up like a 0.5 micromolar solution of hydrogen peroxide, I don't know how stable it would be for. But this is the kind of outgoing quality control that we do to make sure that all the sensors are working before we actually ship them. I may have made a bit of a mistake here because I may have mixed up what you were actually asking for, because I think you were maybe asking for ATP, adenosine triphosphate. We have these sensors obviously on our website, and we also have a calibration solution. Yeah, we do have a calibration solution. Both of these links are underneath this video. So we do have a calibration solution for the ATP. See, I really want this person to come on the workshop because they've got so many questions, and some of them are quite hard and some of them are quite easy. The pH sensor is probably the easiest thing, actually. They've got a pH sensor in a wire format. I must admit, we do test these. I've made them myself. We covered this in workshops. I covered this in workshops on client sites as well. So I can see that, you know, we're testing here, and in this particular example, I can see that this pH sensor was tested at pH 2, 4, 6, and 8. And we do have a calibration solutions for testing our pH sensors. It's quite straightforward, I hope. This is a long answer. Somebody's trying to fabricate immunosensors. They're trying to detect a protein. They're trying to do it probably by binding proteins to antibodies. It doesn't matter if it's antibodies, aptamers, receptors. The story always remains the same. Once they've immobilized the antibody onto the surface, how can they block the surface? They say, well, apart from BSA, bovine serum albumin, I think chitosan is a good other molecule rather. If you don't use a blocker, how are you going to eliminate interference in real samples like plasma? The quick answer is we always use a blocker. Otherwise, I think you're going to, as you already know, get non-specific binding. How do they store the sensor? See, they want to do a shelf life. So if you want to do a shelf life study, you kind of have to store the sensor in the way that you're otherwise intending to recommend it as a product. So my recommendation is to store the sensor in the fridge at approximately 4-5 degrees Celsius. But that's just an opinion. The reason being it's an opinion is because I don't absolutely... Every sensor, every antibody is going to be unique or fairly unique. So I absolutely don't know. I know, you know, you can't really make a product. If your product needs to be frozen and shipped, when I say frozen, if it needs to be like at minus 20 degrees C, it's just not going to be a robust product. So you do have a problem with that. So you have to probably do your shelf life studies by storing them in the way that you intend to otherwise use them. By the way, when people do shelf life studies, they often store them at elevated temperature to elevate the degradation. So you could accelerate the degradation by storing them at room temperature. The problem we've got in biosensors is if you're in the pharmaceutical industry and you're trying to do shelf life studies, you would store your product at 70 degrees C because that really accelerates the degradation. But we all know that antibodies, enzymes, they're all just going to denature at these temperatures. So of course it's going to break down. So it's a little bit harder to do shelf life studies. So in medical diagnostics, especially where you've got antibodies involved. So my thoughts are, you'll probably end up having a product that's otherwise stored in the fridge. So if you want to do accelerated aging studies, or shelf life studies, then you probably should do it at room temperature, assuming you can use what's called an Arrhenius approximation. That let's say the fridge was 0 degrees C, just to make my life a bit simpler. If you stored it at room temperature, let's say 20 degrees C, you will have ended up quadrupling the aging process. So rather than if you did it in the fridge for 4 months, you could do it at room temperature for 1 month, and you would argue that you're in the same place. This is called the Arrhenius approximation, which says that for every 10 degrees, you double the rate of reaction. This is quite commonly used, this assumption, in the pharmaceutical industry and the diagnostics industry. But it's not an easy answer to give you, by the way. I mean, we do this kind of work on large projects with, you know, well-paying clients, you know, and it's not easy. You don't need to necessarily freeze dry or lyophilize. These are really very specific questions that will come out of your project. Thinking ahead, I would hope not. We don't. To be honest with you, I don't think we do lyophilize any of our stuff. We lyophilize for different reasons, but not for this reason. How can you use EIS for quantification without using any redox molecule? You're trying to do what's called non-Faradaic impedance spectroscopy as opposed to Faradaic impedance spectroscopy. You just have to try. There's no way that anyone can really predict whether you'll be successful or not. But I will say one thing: there is Faradaic EIS and non-Faradaic EIS. What you're describing with no redox molecule is non-Faradaic. I mean, you think about impedance spectroscopy. Hopefully you'll get what's called an anapot EIS office, an anapot electrochemical impedance spectrometer, because then I can support you, but I can't support you if you're on somebody else's instrument. You can try it. Most of the time, by the way, we use Faradaic impedance spectroscopy. Whether you may have seen these slides before, but look, be careful. Don't go for gold electrodes. If you can avoid it, don't go for gold electrodes. Gold is really high price. It's $1,900 per ounce. Over the years, we've really tried to get cost out of our gold electrodes. I'm going to finish these slides by just saying, if you're going to use gold, just go for our hyper-value gold electrodes. This is the lowest cost that we are able to make gold at. 625 euros for 100 sensors at 6.25 euros per sensor, which is approximately $6.25 per sensor. I'd still consider it expensive for a screen printed electrode, but it's the price of gold that's the problem here. So hopefully I'll put that link underneath the website. As a side note, hopefully you'll be able to use carbon. I will have a link to a carbon electrode in a minute. Carbon is just a fraction of the price of gold. I mean, gold, people like it because I think it's quite traditional. They make these self-assembled monolayers. They activate these carboxylic acids with EDC, NHS coupling. It's all quite straightforward. And then they can put their antibody into position, and then they block either with BSA, or you can also try chitosan, which I've also covered as well. We do use impedance spectroscopy quite a bit. Here's our link. And this is a protein sensor, and it does work really quite well. Something I'm going to touch upon in a minute. The only reason that ZP is able to make such progress on many of our sensors that we do is, most of our revenue is doing contract development, contract manufacturing people. The only reason we're able to make such good progress is because the electrodes are fundamentally reproducible. Here we got three electrodes tested at 5 nanograms per mil, 3 electrodes tested at 25 nanograms per mil, 3 electrodes tested at 50 nanograms per mil, 3 electrodes tested at 75 nanograms per mil. And it's because the skill of the people in the laboratories, but also the quality of the underlying electrodes. I do have a paper here that talks about using carbon, and my recommendation is actually you do use carbon. I have definitely linked to that paper underneath this video as well. I think this is called Pyrene NHS. It's something you can just get off the chemical catalogs. You can couple the antibodies there, and then you can block with chitosan there. We do use cyclovoltammetry quite a bit during the fabrication process, just to show that we are actually modifying the electrodes. The CV gets distorted as we do the modification. In the paper, we did differential pulse voltammetry, and we did Faradaic. I know you're trying to do non-Faradaic. You can just try the non-Faradaic. There's not much more to be said than that. It's quicker to try and fail than to sort of speculate, to be honest with you. Because if it doesn't work, then you're going to have to go to Faradaic, and that paper would be a good explanation as to how you would actually do it. So I'd rather you didn't go for the hyper-value gold electrodes, but if you so wish, you can. I would rather you go for the hyper-value carbon electrodes. They're just so much lower. Carbon is something like approximately 1,500 euros per ton. So it's 1/32,000th of the price of gold, and it's reflected in the price of screen-printed electrodes. Equipment that you'll need an anapot EIS. You'll need, I hope you'll go for carbon. You'll need a cable to go with the EIS. This is called a LEMO cable, which fits and it's 5-pin, which fits the anapot EIS. And then you'll also need decent connectors. If you don't have decent connectors, you're not going to get decent data out of this entire system. Something I said earlier on is, if you're going to do EIS measurements on an immunosensor, then you're going to need repeatability. One thing you'll notice, we're actually doing a webinar, as I say, in 1 hour from now on FoodSense. When we do that webinar, something that's will be striking to people is that we don't do in-field calibration. When we're really, now a lot of the stuff we put on the web store is just so people can do their proof of concept, their proof of principles, they can get going with their programs. When we're really involved in a program, then we always inevitably try to develop the system to be calibration free. It's really important to try and be calibration free. When you see us do our demos on our in-house products, you'll see that we don't do any calibration, and it all because we've got really good control of our manufacturing. And this is because when we manufacture, we test our underlying electrodes by cyclovoltammetry, just so that we can check out that things are actually repeatable. And then we test across the batch to make sure that our relative standard deviations are not super high. I mean, relative standard deviations on some screen printed electrode products are probably like 50%, 100%. We're keeping ours in the single digits. And we do that because we want it for ourselves. And then our business model is slightly unusual that we also sell them to other people on the web store. So when you'll see with us that, when we're doing products and we're doing signal versus concentration, the only reason we're getting away with this calibration free is because actually of the reproducibility of the manufacturing process. This is pH and oxygen. Somebody's got hold of a couple of our data sheets, one on pH and one on oxygen, and then they also want to know, are we doing any kind of amplification before measurement? The quick answer is we're not doing amplification before measurement. There is amplification happening in the electronics, but this is within the analog front end. There's not like some pre-amplification and then some detection or then some reading of the signal. It's not. Just looking at the pH sensor, there is a data sheet there. Along the top of the data sheets, I've zoomed in here. I can see that this data was gathered at 7.4, this data was gathered at 3.6, this data was gathered at 9.9, 7.4, 3.6, 9.9, 3.6, 7.4, 9.9. So the quick answer to your question is this is the range when making that data sheet that we tested at. Doesn't mean it's the ultimate range. I mean, I've seen some data in this presentation today where we often test between pH 2 and pH 10. So we almost went to pH 2, but we only went to 3.6 when making that particular data sheet. It's a similar question. They also want to know about the oxygen as well. So the way we do this, if anyone comes on our workshop, by the way, everyone gets to make an oxygen sensor and everyone gets to test an oxygen sensor. So if you work for large billion-dollar corporations, I really hope you can have the budget and come on one of our workshops. The solubility of oxygen in aqueous solutions. So if let's say you've got a solution that's being used for cell culture, or you're doing something in the liquid phase, the concentration of oxygen in that solution is probably about 7 to 9 milligrams per liter, which is 7 to 9 ppm parts per million. So what we do is, I could do another video on this. In fact, I think we deoxygenate the solution so we can see the change of signal. So if you're doing cell culture, you'll often have a sort of dissolved oxygen probe, and they'll often say, you put it in the solution and you tell the sensor that's 100%. So that's essentially 100% because it's 100% saturated. In reality, it's somewhere between 7 to 9 ppm or 7 to 9 milligrams per liter, and then we deoxygenate it. And I do have a solution for doing that deoxygenation here. There's a link here. The electronics, everything, I suppose it's worth saying that everything takes place in this board, so it's all shielded up. So essentially there's not some amplification and then it goes to the circuit. The rest of this, by the way, is just a breakout board, so you can sort of make connection with the outside world, but there's no pre-amplification. It all takes place on this, we call it a single-purpose biosensor circuit. So I think in this particular case, this person needs one for pH and one for dissolved oxygen, and I put a link to it there. Question 5, just out of interest, the potassium sensors that we have, you'll find that if you look at our information on our website, we're testing within the clinically relevant range. They were originally developed for testing in blood, and we may put them on our website. But if you're going to be doing human blood testing, you've got to have a proper, well-funded engagement with ZP. You can do proof of principles on, using buffers and stuff like that, but I can't support people using our products to make medical decisions. I can if, we are ISO 13485, but that's really a different engagement. It's not buying something off our website. But that said, we do have a really nice, we have a lovely potassium sensor. If you look up the clinical relevant ranges, you'll find that that's where we're testing at. I've put a link to it. At ZP, we're in quite a position to turn these things into products quite readily, because actually we do have a platform called Senso. Just a quick comment, the potassium sensors are an example of potentiometric, ion selective electrodes rather. It's probably worth saying that we do cover this in our workshops. I do encourage people to come on it. We do have a platform called Senso. Senso is somebody's way of achieving a minimally viable product or actual product in a very short amount of time. So traditionally we've worked with electrochemical sensors. We're also expanding now into spectrochemical sensors as well. Depending on whether it's electrochemical or spectrochemical, there's two platforms for it. They all fall under the Senso umbrella. We do have an app. There's actually an app on the Android Play Store, the Google Play Store, called Senso. I must admit, I don't have a link to it in these slides, but there actually is an app out there. And that app is essentially what that app does is the app doesn't know what assay to run, but it can read a QR code. Once it's read a QR code, it knows, oh, this is this type of sensor, and the app is good to go. So the app tells the hardware what to do. The hardware gives the signal back to the app. The app sends it to Julie, which is a cloud system that I said there were some free workshops at the very beginning of this webinar. Find those workshops, or find the link to those workshops. Come to the Julie one to understand what Julie is. And Julie will then deliver the data back to the person's phone. This happens really quickly, especially in Norway where the internet speeds are fast. So we do have ways. The reason I bring this up is at ZP we can have very high technology readiness levels. You can have a technology readiness level from essentially 1 to 9. When you're first having an idea, you're really at sort of TRL 0, but when you do a little bit of research on it, you're TRL 1. TRL 9 is when you've kind of really done a lot of the background science and proof of principle on it. Now at ZP we also have very good manufacturing readiness levels. You know, for example, our potassium sensors are made on top of a screen printed electrode, so you know the manufacturing readiness levels are high. The reason I bring these slides up is because then people start asking very specific questions about, what about my application? That's an application readiness level. So every application I find is different. People have different scenarios, different products, different price expectations, different user experience requirements. So each application, so I can have a high TRL and a high manufacturing readiness level, but the gap is often the application readiness level. So you have to come to ZP, get the technologies, and then start building out your application readiness levels. I do have a video on YouTube that talks through this a little bit deeper, but I just want people to be aware that even if ZP has a good sensor and good electronics, there's still some work that has to, either you commission us to do or you do it yourselves, and it's quite hard to do it yourselves. Question number 6. This is a simpler one, if I can say, which is somebody just wants to know how to store their lactate activation solution or their sodium potassium activation solutions. For the lactate, we'd rather you didn't freeze and defreeze the lactate activation solutions. It's just we haven't done that kind of study, and I suspect that it will end up denaturing the enzyme. So I'm a bit concerned about it. For our generation 1 sensors, the working electrode is platinum. For our generation 2 sensors, the working electrode is carbon. You can store the sodium and potassium, by the way, in the fridge and just take it in and out as you feel fit. It's a lot more robust. Question number 7. This person had a whole list of, they want to do lactate, potassium, cortisol, testosterone, histamine. You've got to think about the size of your team and the budget that you have. If you're a one-person team with a very limited budget, then you need to focus. And so, you know, if somebody asks me this, I would say, look, the best thing I can do for you is not to support you across 5 sensors, it's just to tell you to focus on one of them. And I would actually say start with potassium. You know, they come in pack sizes of 10. We do have single purpose biosensor circuit, so you've got the electronics sorted out, and you can also get some test solutions as well, so that you can sort of essentially get the sensor, get the electronics, and also get some solutions that you can believe in, and then you can test it. Question number 8. On our website, we have lots of flavors of pH sensors, but on the website they point out, you know, we've got quite strong signal of 51 millivolts versus pH. When they do their own work, they get a signal of 4 millivolts per pH. Yeah, there's something desperately wrong here. Now, what's nice is, I mentioned it earlier on, that at ZP, obviously, I can see what people have ordered. and I can also see the OQC data going out on those sensors. So the guys will have, tested these in the lab before they shipped out. And so I did a quick calculation myself. I could see that the pH and the signal that they got, the pH and the signal that they got, the pH and the signal that they got, the pH and the signal that they got. So I should have really used Julie to do the next part, but I just did it in Excel very quickly. And I saw that, we were shipping out with a signal of 26 millivolts per pH. This is because this was the chloride resilient version, but it's not 4 millivolts per pH. So I don't know why, but we did test the batch before we shipped. So we do a lot of OQC, outgoing quality control. We don't, people who are buying off our web store, these are meant to be quick, low cost, fairly low cost purchases. If people really want to have a deep conversation with us, especially if they're kind of backed by a commercial company, they really need to engage us in a program. But just to say that I can definitely look at your data. Sorry, I just went quiet for a second there because I just wanted to make sure that I seem to, for some reason, lose internet connection just very quickly. The next bit that I want to jump onto is, so I'm just going to repeat myself for a second because I think I might have lost internet connection for a bit. With regards to question number 8, send me photographs and a presentation deck showing me how you do the experiments, because then I can sort of help troubleshoot why you're getting 4 millivolts per pH. It's not right. We have tested the sensors in our lab, and they were good before they went. This is the last question that I'll do today. How to detect glucose and dopamine. Obviously, you can detect glucose and dopamine essentially by having 2 electrodes next to each other. I mean, dopamine you can test by voltammetry. This is not my data, but I'm using it only for educational purposes. You can detect, so this person has not got hypervalue carbon electrodes. I wish they would. If you get a hypervalue carbon electrodes, then you can do dopamine just by doing voltammetry. Obviously, if you're doing glucose, then it's generally an enzymatic reaction. But I would very quickly make the comment that I think you can do these 2 things by really just having 2 sensors next to each other, one for dopamine and one for glucose. But I can understand that they're probably, the question they're really asking is, how do I do it on the same electrode? I've got a super suspicion that if you use what's called a generation 2 glucose sensor, which was linked to one of the questions earlier on when I said it was carbon, you could actually do dopamine and glucose on the same electrode. So you'd have an enzymatic system for the glucose, and you would be detecting dopamine directly on the carbon electrode. I'm not going to say any further how to do that because that's actually quite a bit of work. But the quick answer is, your easiest answer is you can just have a carbon electrode to do the dopamine and a glucose sensor to do the glucose, and just run them in the same sample, but they're actually different electrodes. If you want to do it on the same electrode, I still think you can do it, especially if the electrode is carbon based. But that's really what we call intellectual property, and it's something that we would want to be in a real program to really work that out. So, said a lot. I've spoken for at least 45 minutes. It took me 10 minutes just to get through the questions, or to list out the questions. So I'll just do a quick summary. Materials for CGM. ZP is an ISO 13485 contract developer, contract manufacturer. We do have a lot of materials on our website specifically for CGM developers. This person, question number 2, I have given you some answers. The NO is going to be the hardest. I've also invited you for free to come to our workshop in Norway in November. Building immunosensors. The quick answer is use chitosan. The other quick answer is try and use carbon. And by the way, we do cover the impedance spectroscopy measurements for immunosensors in our workshops. pH and oxygen, I've analyzed those data sheets. I have answered the question of what concentrations of pH and DO there were there. You don't need to worry about preamplification. It's all done on that board. Potassium sensors and blood. Those potassium sensors were originally developed for blood. Please don't use them with human blood. Do your bench testing. It's a web store. It's meant to make things easy for you, but don't use them for medical purposes. The terms and conditions just say for R&D purposes only, unless for medical purposes, which we do a lot of, by the way. Clinical trials, stroke patients, all sorts of people. But those are really proper programs. Keep the lactate sensor in the freezer. Yes, don't get it in and out of the freezer. Lots of times you'll end up damaging the enzyme. The sodium potassium is robust. Just keep it in the fridge. It's fine. Don't if you're quite a small business, maybe just one engineer, don't go broad on the sensors. Go deep first of all, and I say go deep on the potassium sensor. I said here's a potassium sensor. Here's a single purpose biosensor circuit. Here's some test solution that will get you going. Number 8, yes, 4 millivolts per pH is not correct. We did test the batch before we go out. I think I need photographs of your experimental setup to try and figure out what's going on. If I want to co-detect glucose and dopamine, sure, 2 electrodes will do it. One enzyme, one dopamine. Dopamine could just be done on a carbon electrode, like the hypervalued carbon electrode. If you want to do it on one working electrode, that's really something that you would spend your PhD figuring out, or somebody like ZP could do it as well, but I would consider that intellectual property, and I really wouldn't want to discuss it here today. So I will finish with thank you very much. If you have any questions on ZP, don't hesitate to reach out to us. I'm sorry it was quite long this morning, but there was a lot to get through. Okay, thanks very much.